Hello, I'm glad to be with you. Sorry not to be there in person. I'm going to try to make this as quick as I can. I'll be cramming a lot of information in. I actually added some slides that my colleague, Dr. Heather Lombardi, was going to touch on had she been there to talk about those things. But like I said, I'll try to be quick. And if you have questions, I have contact information at the end for you to follow up. So I'm just going to talk today about FDA's, the US Food and Drug Administration's oversight of heritable intentional genomic alterations in animals. So what is it exactly that FDA is regulating? We regulate the intentional genomic alteration, or what we refer to as the IGA, in the animal. That is the specific DNA alteration at each site. But when we say that, that doesn't mean that each individual IGA has to have a separate process and FDA review. There can be multiple alterations that are part of one FDA review. But to be clear, the animal itself is not what FDA approves or makes a risk determination for. We're looking at the IGA, the effects on the animal, the effects on anything that comes from the animal. So our oversight objective is very much focused on safety, to look at safety to the animal, safety to anyone that consumes food from the animal, and also that it does what it claims to do, which of course could have a link to safety, especially depending upon what the claim is, if it has to do with disease or something like that. So I don't have a slide in here about the US coordinated framework, but essentially the US has a policy of each agency that regulates in this area relying on existing laws to regulate. So we have, as Dan said earlier, we do not have a GMO specific law. And FDA, in line with that, relies on its existing law. But we do put out what we call guidance documents that interpret the law and provide specific advice for different types of products. So in this area in 2009, we put out a guidance document on regulation of genetically engineered animals containing heritable rDNA constructs. At that time, we talked about the regulated article as the construct itself, the rDNA construct. We didn't talk about IGAs that were made using other methods, because at the time, of course, we weren't talking about genome editing. But now we are. So we issued a revised draft of that guidance document and clarified that the scope includes all heritable intentional genomic alterations, however they are made. So it can be the older DNA techniques, it can be genome editing, it could be something that we don't know about yet that would emerge. And the regulated article is that IGA again. So we are hoping to put out the updated guidance responding to the many comments that we received in response to the draft. We're currently in clearance on that, and we've kind of split it into two pieces. One that explains the basic oversight framework that I'm going to tell you about in a minute, and one that I'm not going to talk about as much, which really kind of gets into the weeds of the approval process. We've responded to a lot of the feedback that we've received, but we're also going to continue to be open to feedback, and we're issuing that second one in draft so we can continue to refine our approval process in response to stakeholders. So in addition to the approval process, we also do something called exercising enforcement discretion. That is a decision not to enforce a requirement. And that is because, as we all know, no government entity can enforce every single law, every single requirement at all times, because we are all faced with limited resources. In the case of FDA, we have case law discussing enforcement discretion that specifies that it involves a complicated balancing of a number of factors that would include a number of things like agency resources and policy. People may have seen, I mean, FDA uses this quite a lot, actually, and during the course of the pandemic, for example, There were expiration dates that we decided to exercise enforcement discretion over because of shortages, and allowing PPE in that hadn't necessarily gone through the approval process, things like that. But they're all things that we consider on a case-by-case basis, evaluating for that product. So what is our risk-based approach? It is based on the product's risk profile. So in the first category, we have those products that, as in general, we don't need to look at data prior to marketing. The example of this are laboratory animals. There are a whole lot of GE mice that have been around for a very long time. And there are a lot of controls around use of those animals in research. So we have not been looking at those. In the second category are those IGAs in animals that we think could be very well a low risk and therefore a candidate for enforcement discretion. Anyway, so this second category, so those are the ones, there are a lot. I'm not going to have a whole list here, but so for example, somebody mentioned glowfish yesterday. We've gone through this process for many different lines of glowfish. There are animals that are models of human disease that are used in research. And more recently, IgA in a food animal that I will mention also briefly, that exists in conventionally bred animals and has a history of safe use. So then there's the third category, and those would be those that go through the approval process. And I will show you in a minute, those that have gotten an approval. So for the category, as I said, you may have that we're open to looking at low risk, and any developer can come to us and say, I think this product is low risk, and we'll still do that case-by-case review and look at data. So we look at, is it low risk to humans? Is it low risk to animals? Is it low risk to the environment? And if we determine that it is, then we decide whether to exercise enforcement discretion. And if we do, again, we would not expect the developer to submit an application for approval. So most recently, the animal that we made this type of determination for was the slick cattle, which you've been hearing about from a number of different folks. We made a low risk determination for marketing of products that would include food from 2 genome edited beef cattle and their offspring. And we posted a summary of our risk assessment. The cattle have an equivalent genotype to conventionally raised cattle, and there's a history of safe use. So there was talk about off-target mutations the other day. There were some present here, but for us, the issue is, number one, are they present? But then the very important number two is, are there any safety issues there? Do we have any concerns about it? And in this case, the answer was no, they don't present any safety concerns. And these cattle have the same phenotype, the slick hair coat, as conventionally bred animals that have been consumed safely. We also have implemented, I think certainly since the last time this meeting was held, a veterinary innovation program that's available for IGAs and animal cell and tissue products that provide a benefit to animal or human health, food production, or animal well-being. I think we currently have 46 products enrolled. It could have gone up since I made this slide. I won't go into all of the many benefits because we're pressed for time, but there are a lot of them there. And the reason we created this is because a lot of the sponsors and developers of these products are small entities that aren't familiar with regulation. And we want to help them to figure out the process and be able to have the best quality submissions to us that they can, so that we have a smooth review process. So some of the things that are involved with that is that we'll have a kind of pre-review of a submission, so we can flag things that are a problem if there's something missing. But if there is something missing, we will what we call stop the clock, because we have a 180-day review time on particular submissions. And so we'll stop that. And then when the completed piece of that submission comes in, it just restarts at that point instead of having to restart from the beginning of the clock. So there are a number of things like that, and our stakeholders have given us some good feedback that it's been helpful. So this is a list of the approvals. I think, most of them have been around for a while and you're familiar with them. The most recent is at the bottom, which is the GalSafe pig. And as you probably know, that's an IgA that results in non-detectable levels of alpha-gal sugar on cell surfaces. These animals are used for both biotherapeutic uses, xenotransplantation in particular, and also for food. And it's been kind of a nice example. We're talking about consumer acceptance. Right now there's a limited supply for food, but these are available to people who have not been able to eat pork because they have an allergy to the alpha-gal sugar. And, there are Facebook pages of people just going on about how grateful they are for this product, that they can eat pork again, and they really welcome it. As I said, we're sorry we couldn't be there, but I have contact information here for my colleague, Dr. Heather Lombardi, who's the director of the Division of Animal Biotechnology and Cellular Therapies here at the FDA Center for Veterinary Medicine, and Adam Moyer, who leads the animal biotech team. Please feel free to contact any of us if you have any follow-up questions. Thank you.